Hello and welcome to a short episode of Katie the Science Lady. I'm Mrs. Jacobson and today's topic is DNA replication. Let's learn together. Today's quick topic is DNA replication, and this process can be pretty complicated if you get into the really small details of it, but today we're just going to go over a brief overview, talk about why it's important and why it's necessary. So DNA replication, to kind of recap our cell cycle ideas, is the process by which DNA is copied or synthesized. So we're making a second copy of DNA in the nucleus here. It happens during S phase, remember that synthesis phase, and this is because our cell is getting ready to divide and each new daughter cell needs an identical copy of DNA. So we can't just have one copy and split it apart because our cells would only have half of the needed information. We have to replicate the DNA and make a new copy before we can divide those cells. So here we go. Here's our happy little cell. It's got one copy of its DNA. That's enough for one cell, but not enough for two. When S phase happens, it duplicates each chromosome. So you have two copies of each. Now you have enough for each new cell because you're going to split that cell into two. During M phase, the cell splits apart and each new daughter cell has one full copy of the needed DNA or chromosomes. So that's the nitty-gritty of it. DNA replication, we call it semi-conservative, and I'll explain what that means. In essence, it means that part of it is the old DNA and part of it is the new DNA, or the newly built DNA, because each half of DNA is going to be a template. And then you're going to end up with one half of it being the original DNA from before it was replicated, and then the other half of the DNA will be the brand new DNA, or novel DNA. Novel means new. So you're getting half and half, which is kind of nice because you're not stuck with one copy of the old DNA and one copy of all brand new DNA. It mixes together, which is just good because you don't ever want something to just be all old cells or old versions of something. And in this case, it would be all older nucleotides because DNA is made of nucleotides. So if we look at our diagram here, we've got our original DNA. And when it splits, we have both green here. Green is original. Our new DNA is blue. So each copy is going to have half original DNA and half new DNA. This is the most important part of what we're going to talk about. Enzymes are huge for DNA replication. Enzymes are specific proteins that speed up chemical reactions. Without enzymes, DNA replication does not happen. It just doesn't happen. It's going to speed up this process and help it work efficiently because proteins do work. So there's one called helicase, and it sounds like helix, and that's kind of your clue here. Its job is to unzip or split apart the DNA molecule. So the helix unwinds, and then helicase kind of pulls it like a zipper almost and splits those two pieces apart, those two sides of the DNA backbone. Polymerase, or DNA polymerase, poly means many, and it's going to create new things. It's going to add new nucleotides to the templates. So it's making a polymer. That's the base there, polymer, and then ase, it means enzyme. So it is just making polymers. That's its job. It happens at a place called the replication fork. And this is going to be right where the helicase unzips the DNA molecule, right where it splits into two is called the replication fork. And here we have a little picture of it. Right here would be our replication fork. It's been split apart. Now you can't see enzymes in this picture. Normally in another diagram that we'll see in a second, you'll see the enzymes. But here there are no enzymes. But we've got a replication fork here. And then the direction is opposite. So it's going to be adding nucleotides to one strand this way and adding nucleotides to another strand up this way. So that's what we're looking at for enzymes. Those are the two big ones. There are more enzymes that are involved. These are the two you need to know. Here is our little more detailed diagram. Remember, up here was a chromosome. We have a little bit of chromosome action. That coils up into our DNA. So we see our DNA here. We've got all our base pairs in the middle. We've got our backbone on the outside. Here we have helicase, so our enzyme right here, and it's unzipping, and that's the replication fork. Polymerase is this big, almost looks kind of like Pac-Man, like if Pac-Man were trying to eat the DNA. Just because it's yellow, I think I associate it with Pac-Man. And it's adding all these new bases here to create that novel half of the DNA. So it's using the original DNA as a template to follow and create that pattern. So to recap today's learning, in DNA replication, we make a second copy of DNA. This happens during S phase of the cell cycle. And now this happens because we need to have 2 copies of identical DNA to go into our 2 identical daughter cells created in mitosis. We want them to have the same number of chromosomes and be equal. DNA replication is pretty simple. It involves a couple of enzymes. DNA helicase unzips the DNA strand and separates it, and DNA polymerase adds new nucleotide base pairs to create the 2 copies of DNA. Thank you for watching. Please like and subscribe for more biology videos, and I hope you had fun. I hope you learned something, and I'll see you later.